Kia ora, I'm Johnny. I am a PhD candidate also in Kay Hodgson's lab in Monash in Melbourne. Before I say anything, I'd just like to acknowledge the traditional custodians of the land on which I live and work, the Wurundjeri people of the Kulin Nation, and I pay my respects to their elders past and present. And like Paul, I also like to look for structural variants in invasive plants. And I find them all very interesting and fun. So one thing that I think has become abundantly clear over the last couple of days is that no one here needs convincing that invasive species are interesting. And like Paul mentioned, they provide these very good natural experiments to study rapid adaptation. And that's what I have been doing, and we have all been doing. So common ragweed, which is what I'm going to be talking about today, is a particularly nasty little plant. It is an annual hermaphroditic flowering plant. It's endemic in North America, but has become invasive on every continent in the world except Antarctica. It is wind pollinated and it causes really, really nasty hay fever. It's highly allergenic and it's competitive, and I'm very happy that I've never actually had to bump into it in the wild because I also get very bad hay fever. So, as we've heard earlier on, is one of the things that you might want to find or expect to find in an invasive flowering plant is clinal patterns in key traits. For instance, in a flowering plant, you might expect that at sort of higher absolute latitudes, you will have a shorter growing season, and henceforth, there will be fewer days to flower. But alongside this, you might also have a smaller plant. And we have found this, or Kay's previous PhD student, Lotte van Beheven, has found this in ragweed. Berlin populations are tiny when they flower, they're about that big. Whereas some of the more southern southern in the Northern Hemisphere, with longer growing seasons, they will have more days to flower and they are big. They're sort of 1.5 meters or maybe higher. So, knowing this, we've seen this in phenotypic and genetic common garden experiments. We collaborated with Mike Martin and Vanessa Beeker here on producing a whole genome sequencing data set of ragweed across both the native range in North America and the invasives in Europe. And so we did this to 600 plants. 300 of them were modern plants and 300 of them were herbarium specimen plants. So we got a really cool way of looking at the evolution of this thing over both space, but also in time as well. And Vanessa sort of led this analysis into changes in population structure and also microbial diversity. And it's very, very cool. And if you want to hear more about that, you can read that. But what we then wanted to do was then to have a slightly sort of finer resolution look at what the sort of selective processes might be happening across the genome of ragweed. And in particular, wanted to look at parallel evolution and rapid adaptation. And so using Baypass, we, after correcting for population structure, took a overlap between XTX, which is the FST-like statistics, and environmental allele associations, and found that for both of those in North America, there were 1,480 windows, and that was significant. And in Europe, there were 908. It makes sense that there were fewer in Europe because it's newer there and it's been in America for ages and probably more adapted there. But we found that using this weighted Z analysis, 173 of these windows were actually sort of parallel outliers. So that's really cool. We found these sort of like signatures of parallel adaptation throughout the genome. And then bringing in the historic data, we performed a selective sweep analysis by calculating FST divergence in populations between both the modern and historic in that same location. And you can see that, along the genome, there were a couple of very interesting points, and these corresponded to regions in which you saw less nucleotide diversity as well, so it makes sense that would follow with a sweep. And first of all, in Berlin, there's this very interesting peak here, and if you go and look at the name of that gene, look what it's called. It's called early flowering 3. So that's really interesting because that's the kind of trait that you might want to be interesting. So when we did GWAS in across the genome for flowering onset, we found that you can see that peak on the Manhattan plot there is exactly the same location as that selective sweep. And it's really fascinating, especially because you can look at that map there. It's only present in these very northern populations. There's some in Quebec and there's some in Berlin. And when you look at the allele frequency shift in these populations, when you compare it to the sort of null frequency shift of like neutral alleles, the frequency shift of L3 is well off to the right. So it's definitely outside of that null distribution at least. So that was really exciting. But if we go back to that sweep plot here, there's also another really big looking sweep candidate. And this helps me segue very nicely into the next part, which is when I decided, or when we wanted to look for a whole bunch of structural variants in ragweed. So we decided to do that using the same method that Paul just outlined. So using this low struct method, chopping up the genome into lots of tiny little windows, doing PCAs on them all, plotting them out, seeing if they segregate in three clusters. And we found a whole bunch of them which do, and they show the interesting sort of elevated heterozygosity and heterozygotes and all those things, but I won't go on about that too much. But what was really cool in this data is we managed to do this in the contemporary genomes so that we could identify like sort of genotypes of these inversions in the contemporary data, but actually we were able to do it in historic data as well. So we could track how these putative inversions were shifting over time as well as between both the ranges. So if we look onto the far right here, you can see in purple in Europe, the haploblock 448 inversion, which overlies perfectly with that selective sweep, starts off in the historic samples in sort of pinky purple at the bottom here at quite a low frequency, and over time it has gone up to much, much higher frequencies. So that's really exciting, a really exciting thing to see overlapping with the selective sweep, and comparing it again to null distribution of frequency shifts, again it's well up to the side of that distribution. So that was an exciting thing to find, but that wasn't the only inversion that we found. We found a whole bunch of them, and this one in particular was very interesting as well. So we called it HB27B because there's two on this sort of chromosome. And this one here, when you do an association of the homozygotes and the heterozygotes to flowering end in the individuals for which we had phenotypic data, we did find that there were significant associations with flowering end. It also overlaps with a previously identified flowering time QTL. It falls like perfectly on top of the inversion. And I will direct your attention to the North American continent, where again, pink is historic, and the less pink and more green you get, the more modern you become. In North America, we can see these nice sort of clinal sort of patterns in this frequency of the inversion over latitude along the x-axis here. The y-axis is frequency. So that's interesting. There's no significant shift between the two times. Modern and historic are roughly the same. But if you go down into Europe and you look, there is, if anything, declines in the other direction. But as you get closer to modern time, this cline has sort of almost perfectly reformed. And for something that's a sort of associated with flowering time, to see that sort of cline reform is quite consistent with the idea of an invasive species arriving in a new habitat, being maladapted, and then having a sort of cline reform. And it's really cool to be able to like sort of see that happening over time. And I've got a little plot here of the map. You can sort of see in Europe that there's sort of lighter colours in the north, and it gets a bit darker in those sort of southern Mediterranean populations, whereas in the North American one, you can sort of see that pattern there. So basically what we managed to find was signals of rapid and parallel adaptation in common ragweed, both contemporary timescales and also in historic data. We found that inversions underlie a lot of these signals. It was about, for the parallel weighted Z analyses, this overlapped with about 17% of like inversions. were under about 17% of those windows. So that was quite a sort of cool finding. There's a lot of association with key traits that we've found. And we've managed to find that these inversions are not just changing over these sort of spatial gradients in both the ranges, we've also found that they are changing quite rapidly over a small time scale, about 150 years or so. So, yes, I would like to acknowledge Kay, Tim and Paul, fantastic supervisors, everyone who's been involved with this really big project. It's been going for quite a while now. I'm just sort of doing a little role in it. Yeah, so thanks for listening, everyone.
Cool, thank you for that awesome talk. Does anybody have any questions?
That was really cool stuff, thank you. A question about that cline that you showed.
Yes.
I had to write it down, HB27B.
How do I?
It looks almost like instead of a parallel cline, it's almost going to fixation in Europe.
Yeah, the plot's kind of difficult because in Europe the latitudes are higher.
Right.
And it's I didn't really want to plot out the sort of predictions in North America for which we didn't actually have the data, but the sort of shape is kind of the same. If you predict more south than what we have in Europe, that cline does. But yeah, it does look like it has almost reached fixation. If you looked on that map, it was only along the sort of Mediterranean coast where we had maybe some sort of heterozygous populations, whereas if you get deep into its
right, and of course the effect of latitude and climate might be a different slope in Europe than in North America.
Yes.
Right? So if you do it, I guess what I'm wondering, if you do it with environmental variable like season length would be the thing that come to mind. What would that look like?
Haven't done that.
Yeah.
I did in those logistic regressions I did correct for sort of neutral stuff, but no, I don't think we've looked at that in particular. Definitely worth doing that though.
Thanks.
So I'm curious how you defined populations using the herbarium specimens.
Yeah, they were very difficult to do that.
Yeah.
So a lot of the sort of genotyping of the historic ones when I was doing the inversions, that was almost just on the individual level, unless 2 or more individuals were from exactly the same place.
From the same sheet, or?
But yeah.
Okay.
I think I wasn't doing the herbarium stuff, but I would presume so. But yeah, so a lot of that was on the sort of individual level. Yeah, it's something that we've kind of grappled with. I think that's the difficult part of the herbarium kind of analyses, is that population. Yeah. It's hard to sort of genotype a population when they don't really exist. It'd be nice if people made little time capsules, right? And you go dig it up and be like, this is.
Well, I mean, on some plant species, people do collect a lot of individuals per sheet, and I just don't know if that's the case with ragweed. But that. Yeah. So potentially you could do that with some things.
Yeah, I'm not. I wasn't the person doing all the sort of historic
cool
collection. I got the data and looked at it. But yeah.
Cool. Is there anybody else? Any other questions?
I'm just curious, you found a lot of likely inversions. What kind of sizes were you talking about for that in terms of gene content?
Gene content.
Just how many genes?
There'll be medium to large size. Yeah. None was small, but that's not saying there are no small inversions, because I think that low struct or these local PCA methods struggle at the smaller inversion sizes because just less of a signal. But yeah, it's something I'd like to look at, but it's hard to be confident that what you're seeing is a reflection of reality. Yeah.
Oh, it was really cool. Thank you.